I'd like to start by also acknowledging Jordan Briggs, who is a University of Minnesota graduate student who contributed a lot to this study. And I'd also like to start with a disclaimer that this presentation is not about new sources of resistance, even though we're in the new sources of resistance section. It's a study of old sources of resistance, but it has implications for new sources, particularly of adult plant resistance. And so I want to begin by asking the question, why is APR effectiveness inconsistent? And so just a couple of examples of that you can see in the table here. There are two varieties of wheat, Tom and Linkert. These are adapted Minnesota cultivars that were released recently, and you can see their responses to stem rust in both Kenya and Ethiopia over a couple of years. And there's just kind of a hint of a pattern that the adult plant resistance isn't quite as strong in Ethiopia as it is in Kenya. Similarly, you can see an LOD chart to the right of that table showing some mapping that Jordan has done in one of his populations of adult plant resistance. And there's a very strong LOD peak on a chromosome for data from Kenya, but not from data from Ethiopia. So also another hint that maybe APR isn't as consistent as we might think it theoretically should be. And so this kind of gives you the framework of the observations that we had going into this study. And so I have a very traditional outline for this study. I'm going to start with an introduction, and the topic of this talk is going to have to do with Thatcher resistance. And so Thatcher is a wheat variety that was released in Minnesota in 1935, and it has been very important for a lot of the adult plant resistance around the world. So a lot of adult plant resistance is thought to be derived originally from Thatcher wheat. And so we've done some work with Thatcher resistance previously, and that's what I'm going to summarize in this introduction to this talk. And so really the key point to understand about Thatcher resistance is that there is this transgressive segregation. And so though Thatcher itself is quite susceptible in the field, when you cross it with a variety that has the gene LR34 or SR57, you can get lines in the population that have both LR34 and also the resistance from Thatcher that have very strong adult plant resistance. And so previously we used this population between Thatcher and McNeil, 160 RILs, several molecular markers, and also phenotype data from Kenya and St. Paul to try to understand what the resistance was. And first of all, we screened the parents at the seedling stage, and you can see to a number of races both parents are susceptible. However, there are a few races like the race SCC-SC that where Thatcher is clearly seedling resistance. And so we assessed the response of the entire population to race SCC-SC and found that the resistance segregates as a single gene, and that is likely the gene SR12. And so we included the SR12 gene in our map with the other markers using the SCC-SC data. Looking at the response of this population to the stem rust in Kenya and in St. Paul, we found 4 QTL. The most important QTL with the most, explained most of the variation, coincided with the mapped location of SR12. And so this is interesting because SR12 is not effective to race TTKST at the seedling stage. You could say TTKST has defeated SR12, but SR12 is coincident with a very strong adult plant resistance locus in the field. And so we also found QTL on 1AL. We picked up the SR57, LR34 QTL, and then a small QTL on 2BS. The other thing I want to explain about what we previously did is that not only did we see these different QTL, we saw this interesting interaction where many of those genes, particularly SR12 and 1AL, the QTL on 1AL, had an epistatic interaction that explained a lot of the variation in the data. And so what that means in the field on those lines is that we observed that the mean of rills that didn't have any of the QTL had a stem rust severity around 65 averaged across those different environments. If you had any one of those single QTLs, you didn't get a significant reduction in the severity. But if you started adding multiple of those QTLs together, then you saw a significant reduction. And so we call this type of interaction complementary epistasis. And it's interesting not only because of this, but also because it involves this SR12 gene, which is considered defeated to race TTKST. And so just a summary of that introductory information that's already been done previously is that there is complementary epistasis in Thatcher that explains the adult plant resistance, and that a defeated resistance gene SR12 coincides with the adult plant resistance. And so we thought that was a very nice start into understanding the adult plant resistance in Thatcher, but we have additional questions. And so one of those questions that forms the objective, the specific objective for this talk is adult plant resistance in the Thatcher-McNeil population race specific? So moving on into the materials and methods, what we did to try and address that objective, we took the same population and then we assessed the population with 4 races, North American PGT races in the field in Rosemont, Minnesota, over 3 years. And then we also assessed the population with the same isolates of those races at both a cold temperature regime at the seedling stage and a high temperature regime at the seedling stage. The reason why we did that is because we know that SR12 is temperature sensitive, and we wanted to be sure we could pick up SR12 if by chance maybe there's some relationship between the seedling data and the adult plant data. So looking at those 4 races that we selected, one of those, race QFCSC, is avirulent to SR12. And so we wanted to kind of see what the resistance in Thatcher looks like in the field to an SR12 avirulent race. However, the other 3 races are virulent on both of the parents. And so the observation of adult plant resistance to those races would be indeed that, informative of adult plant resistance. One thing that we did in order to analyze our seedling data is we converted our Stakeman infection types to a 0 to 9 linear scale, and that was simply so that we could use statistics and analyze those data. The Stakeman infection types are very much qualitative responses, and so we converted that to a semi-quantitative scale. And so for the rest of the talk, you'll see the infection types as the converted infection types on the 0 to 9 scale. So what do these nurseries look like in Rosemont? And you can see that from this figure. You see the orange lines indicating where the entries are planted, and then the black lines show the spreader rows, which are planted perpendicularly to the 1 meter entry rows. We also planted these 8-foot wide strips of just a common variety, and that was useful for us because we contract out the application of herbicide to the University of Minnesota, and so it's easier for them to just drive a tractor with a big sprayer boom to apply the herbicide. And so that makes it a lot easier on us. The nurseries were sprayed, the spreaders were spray inoculated 2 to 3 times with a single PGT isolate. So we had 4 of these nurseries in Rosemont every year, each inoculated with a single isolate of Puccinia graminis. Here's a map of where those nurseries were located. You can see that there's kind of some overlap of where a nursery might be in one year to the next. And one thing I'd like to point out is that in 2014, we weren't able to get nurseries of QFCSC and also QTHJ. So we only have the 4 races in 2013 and in 2015, and in 2014, we only have 2 of the races. And so Rosemont, Minnesota, you can see the edge of the city here. The nurseries are at the University of Minnesota station at Rosemont, and in any given year, the nurseries are each spaced at least 500 meters from each other. Here's a couple pictures from those nurseries. What we did is we thought it was very important to try and maintain the purity of the infection in these nurseries. So the same person did not visit more than one nursery in any given day after inoculation. And then we recorded severity and infection response twice for every entry that was replicated twice in the nursery each year. The severity was recorded on the 0 to 100 modified Cobb scale. Infection responses were recorded as usual and then converted to a numerical scale according to Rolfs et al. In order to calculate coefficient of infection. We calculated coefficient of infection to best estimate yield loss. That's what we're really interested in studying stem rest, is its effect on yield. And so for the rest of the talk, when I talk about the field data for stem rest, I'm going to be referencing coefficient of infection. And so the last part of the materials and methods, unfortunately, is it's very important, but it's also kind of boring. And so I'm just going to run through it really quick. We used a function in R in order to transform the data to best fit a normal distribution for further analyses. We used ANOVA to estimate the fixed environment and race effects. And then we used the random or line effects and the residual effects in mapping. And then we used our previously constructed map. And first, we completed composite interval mapping to identify the QTL, follow that up with multiple interval mapping of those QTL to really resolve the effects of those LOD and the interactions among the different QTL. We used Tukey's honestly significant difference test in order to assess differences among QTL means and different trait means. So results. The first thing I'd like to show in the results has to do with the seedling data. And so you can see here a density plot, which is like a histogram. On the x-axis is the 0 to 9 converted Stickman infection types, and then the density of those. You can see a clear bimodal distribution, which is exactly what we would expect because race QFCSC has the gene SR12, which is effective. QFCSC is avirulent to SR12. And that experiment was done at the cold temperature. And at the higher temperature, we found that that distribution changed dramatically. We still had lines with a very high level of resistance, but there weren't as many, which is also what you would expect because SR12 is temperature sensitive. This is an important slide because what it shows is the response of the parents, Thatcher and McNeil, to QTHJ, which is virulent to both of the parents. But you see in 4 of the RILs, the typical susceptible response that's very common in the population, but we did see some seedling infection types that were lower. And so this is interesting because it suggests that there's also some transgressive segregation for seedling resistant, which is very interesting. And when you look at the entire population to race QTHJ, you see that the vast majority of the population is quite susceptible to race QTHJ, but there are a few individuals that have lower infection types. And so an infection type of 6 corresponds to around like a 2+3 Stickman infection type. And so most of the population is clearly high, but there are a few individuals that seem to be intermediate. Similarly, for races TPMK and RCRS, most of the population was quite high, but you have a few individuals that are low, and especially at the low temperatures, you have this tail that goes out with a few individuals being intermediate. So in the field, here's the data for coefficient of infection for race QFCSC. You see the bimodal distribution again, which is what you would expect for an avirulent isolate. However, in 2015, the response was quite different. In 2015, the stem rest was not as heavy as in 2013. In QTHJ, the coefficient of infections in the field showed mostly susceptible well, not really susceptible, but just high coefficients of infection and then lower coefficients of infection in 2015. Similarly, the pattern held true for the 2 races where we had 3 years of field data. And you can see that there's very large differences across year, and maybe you could say that there's some differences among the races. So to really look at what's going on with those data that we observed in that format, I'm going to display in this table the Pearson's correlations of coefficient of infection. And so the dark green corresponds to higher correlations, and then the I guess in this format it looks kind of like an orange or brown is the lower lower correlations. And so the main point is that in any given year there were high correlations among the different coefficient of infections observed in the different nurseries. And then for example, race TPMK was highly correlated with race TPMK in 2014 and also with race TPMK in 2015. And so the same race was highly correlated with itself in different years. And then the least correlated races were race RCRS and QFCS. And so we saw that pattern in 2013, also in 2015, that was the lowest correlation observed. And what's also, I think, quite interesting is that variation that we observed at the seedling stage, that was mostly variation in susceptible responses, you know, lines with 3+ or 3+, or 3, that variation corresponded very strongly with the variation that we observed in the field. And so these correlations are actually, you know, resemble some of the year-to-year correlations of the same race of field data. And to look at what that means for different QTL, I'd like to put up this graph, and this is the QFCS field coefficient of infection data, and there are 6 different chromosomes that are highlighted, and these are 6 chromosomes where we found significant QTL with composite interval mapping in one of the traits. And so just QFCS is displayed here. You can see the very strong peak corresponding with SR12, which is what we would expect. Also, in the seedling data, the seedling data mirrored the field data. And it's kind of difficult to look at all of these composite interval mapping graphs, so I'm going to skip that and just show tables, which I think is a little bit easier to see. And so these are the results of the multiple interval mapping just for race QFCS. You see the LOD significance of SR12 being very high. The darker blue shows a higher level of significance. And then you can see in 2013, LR34 was not very significant, but in 2015, LR34 was very significant. And so for the other races, those are displayed to the right. You can see that even for QTHJ, which is technically virulent to SR12, SR12 had a very, a QTL linked to SR12 had a very high LOD. And then you also see this pattern where LR34 is more effective in 2015. With TPMK, you still see significant QTL for SR12, but it kind of is less. And for race RCRSC, the QTL in 2 of the 3 years are non-significant. And again, in 2015, LR34 was had the highest significance in 2015. And we did see significant interactions across some of the QTL in certain years. Another way to look at those data is to look at the additive effects. And so this graph shows the effects of coefficient of infection. And so at the bottom, you can look at the mean coefficient of infection of the population observed for those different traits, the percentage of the genetic of the variation that was observed explained by this model. And then you can see the alleles conferred from Thatcher reducing coefficient of infection, and then the alleles from McNeil that were increased the coefficient of infection. And so the take-home message from this slide is that there's really 3 big important QTL that were the most significant. SR12 1A and LR34, and those were the most consistent across the races. And so to kind of visualize what these data mean on these lines, I'd like to show this set of charts. And so you can see the average coefficient of infection across the different years. In this case, for race QFCSC, this is a very simple story where reals with SR12 or reals with SR12 and other QTL were highly resistant. And if you didn't have SR12, then you were more or less susceptible. The picture becomes less clear when you look at the other races. So first, for race QTHJ, you do see that SR12 did have a significant reduction, but you really needed SR12 plus other QTL to get a really strong resistance response. For race TPMKC, there seems to be an increase in the susceptibility overall. And again, if you had all of the QTL, you did okay. And then finally, for race RSC, you see that the QTL means really weren't that great at all. And even if you did have all three of these strong QTL, you didn't reduce coefficient of infection that much. And so it seems like there's different levels of effectiveness of these QTLs. And so to try and tease that apart, I'd like to zoom in on the RILs that have that SR12 plus 1A plus LR34 genotype. So these are those 17 RILs here. And here is their average coefficient of infection to the different races. And so you can see the mean coefficient of infection to QFCSC is quite low. And that makes sense because QFCS is SR12A virulent. But for the other three races, you have a different order of magnitude of resistance. And to try and quantify that in a statistically significant way, well, first let me also show the genotypes of the QTL for the other three QTL are displayed over here. And you can see that there really isn't a great easy explanation where lines with the highest resistance didn't necessarily correspond straightforward with the McNeil resistant allele for those other QTL. And so again, to try and resolve the significance, what we did is to quantify the reduction of coefficient of infection conferred by that gene pyramid. And so, for example, for race QFCSC, we want to see what is the proportion of reduction in coefficient of infection. And so those results for 2013 are displayed here. You can see for the SR12 avirulent race that the reduction was quite high, over 80% reduction. But for the virulent races, the percent of reduction was quite variable, where QTHJC had a high reduction, whereas RCRSC had a very low reduction. So you could say, well, maybe that's just a function of where those fields were that year. And so it's important to look at multiple years. And so for 2014, we only have these two races. We don't have a significant difference, but we do have a large difference between the two means. And then again, in 2015, we do see a significant difference among these three virulent races. And so those are the results that we found. To kind of bring that all together into a few conclusions, the first is that we did see substantial variation over years. And so this is not unexpected in any way. Many people who do QTL mapping find that in their studies. We also found that variations in what you might call susceptible seedling infection types correlated very strongly with field coefficient of infection. And so this kind of begs the question, would it be possible to predict APR using seedling infection types? And so to kind of test that hypothesis, we ran that population also at the cold temperature with race TTKSK and mapped those data. And then you can see the distribution of those data here. The population is very susceptible to race TTKSK, but there is a little bit of variation. When you look at the lines with the lowest seedling converted infection types, those lines have these QTL genotypes. And so picking these lines that have like a 3 infection type or a 3- infection type is very predictive of adult plant resistance QTL in the field, which we thought is quite interesting. Most importantly, how did we address our objective of is adult plant resistance in Thatcher McNeil population race specific? And I'd like to conclude with this take-home message that the data suggests that the effectiveness of wheat stem rust adult plant resistance varied across PGT race tested. And so here below are just the p-values for the TPMK RSCSC comparison. You can see that there's a consistent difference between those two races in the resistance conferred by that specific gene pyramid. This suggests that adult plant resistance is not the same for all races, at least for this population. And I'd also like to conclude by saying that this population is very much removed from reality in some ways because it's an old cultivar crossed with a very susceptible modern cultivar. None of the lines can be used in breeding. We use this population specifically because of the susceptibility. And it would be very important to validate this type of hypothesis in more agriculturally significant material. And Ndale Hailu is giving a presentation later today that is looking at some similar types of data from Ethiopia. And so it will be exciting to hear his presentation from Ethiopia. With that, I'd like to acknowledge several people from my lab: Sam Stockton, Jerry Ochoki, Kelly Sidla, Arena Ide, Garima Singh. This research was funded from our CRIS project, so it was funded by the US taxpayers. And I'd like to thank the University of Minnesota Outreach Center at Rosemont, the Minnesota Supercomputing Institute, and also Liang Gao is a postdoc with Jim Anderson, and he wrote a Perl script for converting these infection types and infection responses, which is very helpful in generating these data quickly. Thank you very much for your attention.